Fantastic. So our next speaker is Mahan Ghafari, and he's going to talk to us about SARS, determinants of SARS-CoV-2 evolutionary dynamics in persistently infected individuals. Oh.
Good morning, everyone. And thanks to the organizers for the opportunity to speak here today. I'm Mahan. I'm currently a postdoc at the Pandemic Sciences Institute at Oxford, but I will soon be forming in my own mini group, the Department of Biology at Oxford. And today I'll be giving almost a follow-up talk to the previous fantastic talk we just heard on within-host evolution of SARS-CoV-2 during persistent infections. And why are they important? Well, we believe, and I think some people in this room might disagree, but that these are the most likely source for the emergence of highly divergent variants of SARS-CoV-2 like Alpha and Omicron. And when we think about modes of SARS-CoV-2 transmission, we often think of it in the top panel. Let's see if I can get the cursor, laser, okay. When we think about modes of transmission of SARS-CoV-2, we think, typically acute infection lasting for a week or two. The virus may accumulate 1 or 2 consensus changes, goes through a tight bottleneck, the virus gets passed on to the next individual, and you have this established sustained transmission going on in the population. So we would expect typically 1 or 2 substitutions per month. But then there's this less likely path that the virus may cross, and as we've shown is actually rather prevalent in the population, where the virus may persist for longer. They may be immunocompromised, but there could be other conditions too which allows for the virus to persist. And as a result, under some conditions, the virus may end up accumulating a lot of mutation, a lot more, to the point where it turns from blue to pink, so you could tell it's mutating. Anyways, and then so this was the within-host picture. The between-host picture, this is actually not shown properly. This was supposed to be blue, this was supposed to be pink. But anyways, you have this, when we think about the within lineage rate, is that chains of acute infection, that the virus gradually accumulates mutations, but then there's this saltation, abrupt jump in evolutionary distance, which I would call the between lineage rate. And that is what we believe is dominated by this sort of chronically infected individuals acting as a source. So this saltation would then result in the pink variant emerging, and then you could have a sustained transmission within that lineage as well, which would give you another within lineage rate. But then that saltation is really coming from the non-synonymous substitutions, but the synonymous within or between lineages are pretty much the same. So it's adaptive. I'm an evolutionary biologist, by the way. A lot of vocabulary I'll be using is evolutionary. So the research questions we set out to answer were really revolving around 3 points. Are there any host characteristic associated with being persistent infections? Is there variation in viral within host viral rates between individuals and across the genome? And are there any factors associated with elevated viral rates? So this is work that we recently put out as a preprint. You're very welcome to check it out. Just briefly on this data that we had, this is part of the ONS COVID infection survey, arguably the largest surveillance study anywhere in the world, which we collected samples of individuals from April 2020 to March 23 on a weekly basis during the first month of the participants enrolling, and then monthly thereafter, regardless of whether they presented symptoms or not, right? So, and it was representative of the UK population. And as a result of that, we collected 135,000 viral genomic samples from about 100,000 individuals over that 3-year period. And pretty much since the emergence of Alpha after, around December 2020, pretty much everyone with high viral load, that is CT below 30, had their viral samples sequenced. So the way we defined persistent infection, and as we recently shown, about 3.5% of all SARS-CoV-2 infection can last for 30 days or more, and that is any individual with 2 or more positive PCR samples that have higher viral load and are at least 26 days apart, and the sequences belong to the same major lineage of the virus, say BA.1. And the key criterion here is that these sequences should all have a single nucleotide polymorphism relative to the population level consensus that is shared across all samples from that individual and is considered to be rare in the population. So only these individuals have it, but not the rest of the population. So based on those set of criteria, we identified 576 persistent infection. Majority had 2 samples collected, but there were quite a few with 3 or more samples. The longest one had 8 samples over a span of a year. Most of them came from BA2 and BA5 infections, but we also had alpha, delta, XBB as well. So the first thing we found was that these persistent infections were overrepresented predominantly in the elderly population above the age of 60, and also in males that were made 56% of the whole cohort. When we compared it to the background of acutely infected individuals in each sex, shown here in gray bars, that's the background. And so yeah, they were predominantly in the elderly. Then we tried to investigate the within-host evolutionary dynamics of the virus, and the way we did that was to identify de novo mutations. So these are new mutations that pop up during the course of the persistent infection. And we used the changes of the allele frequency as a proxy for measuring genetic distance. So orange here in this non-synonymous, and green is synonymous substitution. So over time, you see a very clear and nice clock-like evolution going on, where you have this gradual accumulation of mutations over time. And as you could see, majority of these changes are coming from non-synonymous. As the previous speaker said, that's because they make up 80% of the mutations in SARS-CoV-2. So what was interesting was when we looked at across all of our 576 persistent infections, we saw quite a bit of variation in the genome-wide evolutionary rate, and that was pretty much dominated by the variation we saw in the non-synonymous changes, whereas the synonymous rate was pretty much consistent. And if I put my evolutionary hat on again, this makes sense because you think of synonymous as being roughly neutral, so there is not much variation. It acts like a neutral clock, if you like. The synonymous rate is pretty much consistent around for every individual, but there's more variation in non-synonymous. Those are changes that can change the protein and whatnot. It had a nice, almost bell-shaped curve, but then there's this long tail towards the right, where some individuals seem to be highly, adaptive or evolving very rapidly, basically, the virus within them. So going back to that picture I showed you earlier, this between lineage rate for SARS-CoV-2, we wanted to see if we can associate it with these sort of accelerated within-host evolution during persistent infection. And when we sort of did that for many of the SARS-CoV-2 major lineages, we found that the between lineage rate is about 1 × 10⁻³ substitutions per site per year, but actually turns out only about 10% of our persistent infections have evolution rate as high as that. So the first message here is not every persistent infection would have an accelerated evolution rate. There's variation, and not all of them would lead to production of highly divergent variants, only a smaller portion. Then we try to investigate whether the evolutionary rate is associated with any host factors such as vaccination status, prior infection, sex, age group, viral load dynamics, or virus lineage. We didn't find any association. But we did find that longer lasting infections tended to have higher evolutionary rates, which was interesting. Then we looked at the variation in the evolutionary rate across the genome. I know the next speaker is going to talk about, it's not all about spike, and I would agree with that. Nevertheless, there was an accelerated evolution going on with the spike, dominated by non-synonymous. So if I zoom in on that, you see particularly accelerated adaptive evolution in the N-terminal domain and receptor binding domain. Not surprising, these are the hallmark regions of SARS-CoV-2. All of the hallmark mutations of major lineages come from that. And what was also interesting, we found there was a lot of recurrent mutations in there too. A lot of, in fact, we found in some cases, 16 de novo mutation happened in 16 different individuals during the course of the persistent infection. So if you look at across all of our individuals, again, those recurrent mutations that is found in 2 or more individuals, half of them had a positive fitness effect in the population. So they were beneficial both within host because they were recurring, but they were also beneficial at the between host. And they were actually mostly coming within this spike. But guess what? The other half that were not positive between host, but recurring within host, they were actually probably outside spike. Mostly they were coming from ORF1AB. So that was also another interesting observation, suggesting there's this sort of competition of transient fitness advantage within host, but then doesn't translate into between hosts. So some mutations are unique to persistent infections, basically. So just to wrap up, being male and above the age of 60 is associated with higher risk of persistent infection, and nearly half of the recurrent mutations we found are unique to persistent infections and not rarely seen in the population. There is variation in the within-host viral rates, and that's dominated by non-synonymous substitution, particularly elevated in the N-terminal and receptor binding domains. We didn't really find much of an association with host characteristic, but we did in fact find that longer-lasting infections may experience higher evolutionary. And with that, I'd like to thank Katrina Lithgow at Oxford and her team, and ONS COVID infection survey team led by Sarah Walker. Thank you all for listening, and thanks for the funders.
Wonderful. Any questions? Oh, lots of questions. Lovely.
So, lovely talk.
Thank you.
Lovely talk.
Thank you.
The question I had is, in that 10% of cases where there was a higher mutational rate in persistent infections, did you ever look at the patient to see, like, genomic-wise, whether there was any characteristics specific about those 10% people, or even like from like a disease point of view, like was specific characteristic about them?
That's a great point. Short answer is no, not yet. But basically, we don't have patient data. So we have certain metadata like age, sex, and whatnot, but we really don't know their underlying condition, which would have been really great if we did. I mean, that data exists, but we cannot access it, basically. So yeah, there's a limit to how much we can push this, basically.
Great talk, Mahan. I just wondered if you, it's similar to Fed's question, did you have treatment records to stratify your data by?
Sorry, say again.
Did you have the treatment records, so the drugs that they were given?
That's what I was just trying to say. Yeah, we don't know, but I'll tell you one thing. We do see signatures of certain mutations. So like I told you yesterday, we do in fact believe there's one individual who is probably on molnupiravir. There's quite a few on other antivirals, just based on the mutational signatures we see.
And were they the patients that, with those signatures that had the increased evolutionary
viral rates? Yes.
Yeah.
Let's go for one there, and then Julia.
Nice talk, thank you. Excuse me. Yeah, when it was like you showed a little graph at the beginning of the number of like patients, and there was like the male 60 plus, but then at the bottom there was like, why do you think there was like a smaller peak for like the younger, like under 20s?
That's not as statistically significant though.
Okay.
It's just small numbers. I'm trying to find it. Yeah. So
Yeah.
it's just small numbers that you can't establish statistical significance that there's lower representation of youngers.
Julia, and then Bella. No, Julia first, then Bella.
It's great though. I was panicking that no one would ask me.
I'm going to record that, Julian first, then Dallin. I like that. So that was, that was beautiful work. So I'm going to be the skeptic in the audience, right? So when you look in really, really healthy people, you can still see 13 to 14 non-synonymous changes occurring in them across the course of infection. So I would say, what is more likely to generate a new variant? The transmission of a virus going through multiple people over the course of, say, the 60 days of someone who is persistently infected. Because if we posit that the virus going through the immunocompromised person is not in a landscape with immunity, and it somehow is then spread from that person into someone with a fully functioning immune system, surely it's going to be at a fitness disadvantage.
Thank you for that. That's
that's the skeptic.
No. I'm glad you brought it up. I was expecting someone would say something about that. Thank you for that.
David Matthews at the end.
No, so I'll distinguish. I'm trying to be careful here. So you have chronically infected individuals immunocompromised. These are probably not all of them, right? So these are from the general population. And actually, we do have large cohorts of immunocompromised. I looked at across the mutational signatures, and these are different from the mutations that they acquire. But to your point, I would agree, it's a bit tricky, the landscape should surely be different, but then I also recently looked at this sort of evolutionary dynamics. We published a paper in Frontiers in Virology you might want to check out, but basically we found that, something that could happen is you have this chronic infection going on for a long time, virus accumulation, but then it might trickle into the rest of the population at, very small amounts, and then those may carry over those extra mutation you need to then boom, So yeah, it could be more nuanced, and thanks for bringing that up.
Last one from Dallin, and then we'll move on.
I think a small question was, are these all nasal swabs?
Yes.
Right, so I guess there's a question about where else the virus is persisting.
That's a great point. We do, in fact, find this. So in our Nature work, we did document about 30% of these individuals shown rebounding viral loads. So there was some intermediate time where nasopharynx was negative, basically. And that does suggest that the virus might be involving in the gastrointestine or somewhere else, and that's been fairly well documented in many cases. So we do believe, and we do have evidence of that also happening, where we see a lot of minor variants popping up at one point, boom, disappearing at the next time point, suggesting subpopulation popping in, popping out.
I guess my second one was more of a comment, really, was when I've seen these sort of discussions about in-host evolution, it's always sort of virus by virus. And I just wondered if you'd considered comparing this to an established coronavirus that's been in the population for a long time, because I think it's just worth highlighting in this context. This is a bat virus, not a human virus, right? And whether or not what you see in a chronically infected patient is something to do with adaptation to humans, or at least there's that added driver, and what a more established coronavirus might look like in that same condition.
Yeah, that's a great point. I don't know if I have a good answer to that, but I'll leave you with this thought of, I've been thinking about this problem, and I wonder, can we even say chronic infections could be a common mode of generation of variants, not just in humans, but generally for viruses? Does this same stuff happen in flu? Does the same stuff happen in bat? You're nodding probably not.
Gut pathogens in bats.
Pardon?
These are commensal gut pathogens in bats. Probably persist for much longer than the acute period that we're talking about for respiratory infection in humans. So I think if these are chronic infections that we're talking about in the gut, that may be more like the niche that they're persisting in the bat. Right. So if it's a gut pathogen, if it's becoming a gut pathogen in people, you may be recapitulating more what's happening in the bat.
Yeah, let's follow up on that afterwards. Thank you for
I think we need to carry this on at coffee break.
Yeah.
Thank you very much for that. And the next talk, thank you. Oh, sorry, thank you.